presentation on stroke proteome analysis. So stroke is the sudden lack of blood supply to the brain, and most strokes are caused by the blockage of arteries leading to the brain, and this is called an ischemic stroke. So I was first interested in looking at endothelial cells because those are the cells that line all blood vessels, including arteries. And I was also interested in looking at two other cell types, platelet cells, which are cells that help form clots, and peripheral blood mononuclear cells, which are peripheral blood cells with a single round nucleus, and to see how these affect stroke and how they're affected by stroke. Using proteomic analysis, I could find potential biomarkers for stroke and identify the cell type that it originated from between endothelial platelet and mononuclear cells. Biomarkers are important to correctly identify and diagnose the ischemic stroke patients, to give the right treatment to them, and to remove the blood clots and restore blood flow. So with further study in clinical trials, accurate biomarkers can be set. Thank you.
So hi Shreya, you did a really good job. That was a really good analysis, and I really liked how you explained your approach to things and how you had figures for every slide. I mean, that's really important when you're trying to present your work and try to explain what you're doing, is always to have like a figure or a pathway to explain. So that was really nicely done. And I understood what you were doing, what was the problem that you were trying to solve. And it was laid out really well. So I have a question for you. So now you've come up with this set of 7 proteins, correct? Some of them are in your platelets, some of them are in endothelial cells. What do you think what do you want to do next with this set of genes? Like,
I think that researchers could perform clinical trials on different patients to ensure that these biomarkers could be universal and an actual way to determine if a stroke has occurred. So clinical trials would be a good way to ensure that.
Yeah, that's a good answer.
Hello all. It's been a really nice ride for me to have mentored these students through 8 weeks, and they have all put in time and effort into this project, and they have hopefully learned a lot. They had all started at starting level of biology, and now they can talk like graduate students. So that's very encouraging for me. And one of the stuff that actually came out of Shreya's work, which is IGF-1, which was found to be highly enriched in endothelial. It's usually a hormone growth, insulin-like growth factor hormone that's produced by liver cells, as far as we know. But her work identified that as a potential biomarker that is possibly enriched in endothelial cells after having compared mouse knockout data. So potentially Shreya's work actually identifies that it could be a biomarker for stroke. So that's something, it's a learning process for me also that happened while this analysis was being conducted. So I was, it was a real pleasure to mentor these students too.
This is my cardiovascular disease research, and this is focusing more on diabetic endotheliopathy and how it is connected to diseases in the cardiovascular system. Okay, so first off, what is the goal of this research overall? So my ultimate goal is to be able to identify the secreted proteins that come out of the cell and to mark early endothelial damage. Basically, when we look at this early endothelial dysfunction, we can detect diabetes in a patient and further prevent other cardiovascular consequences. So next thing is having a brief overview of diabetes. So it is important to know the difference between type 1 and type 2 diabetes in this research. And to begin, type 1 diabetes is more of a heredity and genetic cause of diabetes. And this form of diabetes is located within certain genes that are passed down to offspring. So my conclusion is that early endothelial dysfunction is an early sign of diabetes. And if we locate these properties, the secretion properties of the proteins within the cell, we can possibly mark that there is an early sign of diabetes, which can then help prevent cardiovascular disease. And the reason that the type 1 and type 2 diabetes didn't have common proteins was because the research paper 1 was on rats and research paper 2 was on humans. So they will have significantly different sets of results, and there won't be many commonalities. So thank you for your time.
That was great. That was amazing. And your art, I mean, if in the future you want to have a career in science illustration, it's a really good career to have. And it looks like you're really artistic. So that might be something to consider. Really good presentation. I really liked, again, how you set it up and all. The main thing, I think, in research is because there's so much data and there's so much of information. That you want to break it up with images and all these creative images that kind of catches the attention of the people listening to you. And it's a technical question about your paper 1 that used rats as a diabetic model. Do you know, just from the details, whether it was a diet induced diabetes, or was it a genetic rat model?
So the paper 1 was genetic because all the rats had type 1 diabetes, which is a genetic disease and genetic type of diabetes. So yeah, they collect, and they also did a few environmental facts. They put the rats in a hyperglycemic environment, which basically triggered that type 1 diabetes. So it was, I would say it was genetic.
All right. Thank you. Great answers.
Well, not the question, but yeah, I agree. In this case, the research was really complete and many of these levels. That's really important on the details. Thanks. And the graphics in this case, yeah, is beautiful in this case. The way you describe the diagrams, etc. Probably my only question will be if you can think in this case, if you cannot find a really good biomarker on this level, on this point, can you think what will be your next step to try to finally better data or another experiment? I don't know, you are thinking about that.
Yeah, that's a good question. So if we're not able to locate any endothelial dysfunction, or if that's not one of the biomarkers that really shows us that a patient will have diabetes, then I would just say that there should be an experiment that collects a larger number of patients than that was used in the experiments that I had, because the papers that I used had a very small sample, so it may not be, it may only be accurate for that sample of patients. But if we took a large amount of people, maybe we can see a commonality within the functions within their body for those who are diabetic.
Yeah, exactly. I agree with you in this case. You have a larger population, you have more chance to have something that is in common, because as you say, this is multigenic. So it's really hard to find the right biomarkers on this level. Right. Well, thanks.
Thank you.
Thank you. So today I'm going to be talking about the regulation of Rho GTPases and their role in aortic stenosis. So first off, I'm just going to define a few terms. What is angiogenesis? Angiogenesis is the process in which new blood vessels develop from pre-existing vessels, and it's what forms our veins, capillaries, and arteries. Next is what are endothelial cells? They're basically the barrier between the vessel and the tissue, and they regulate movement and the flow of fluid into and out of blood vessels. 0.59. Lumican is found in the extracellular matrix, which again confirms the conclusion that the ECM proteins are majorly responsible in leading to aortic stenosis. How does this connect to the Rho GTPases that we discussed in the beginning? Well, in previous studies, there has been a connection between Rho A and Lumican. It was found that fibrotic changes it has been found that fibrotic changes show that Rho A overproduces Lumican in the eyes, and this indicates that there is a possibility that Lumican can be regulated by Rho GTPases, confirming the connection between the regulation of Rho GTPases and their role in aortic stenosis. But of course, further investigation has to be done to find this connection clearly. And that's pretty much it. Thank you.
And your presentation skills were really good. You were really calm, and I liked your whole setup and explaining the problem and the issue. And so one thing that I would definitely highlight is I, as a researcher, actually study stenosis. So this was really interesting. And So this is just goes to show that even a simple research that you've done, how it can, translate into something that I can think about and use. So thank you for that. You actually kind of opened my eyes to a few things. And it's a very important question about, and I do agree with your analysis about the ECM proteins, because when you study anything related to the heart or even the aorta, it's really important to understand the ECM proteins and the role they play in making, in basically saving the integrity of the heart and the structure of the heart, and how it can lead to fibrosis or control fibrosis. So it was a great presentation. So what are your next steps once you have this piece of information?
I think that it would be important to go and like check specifically the transcriptomic regulation. So like how much of the mRNA is really being formed for the Rho GTPases, and in like in the ECM, I think like when it comes to aortic stenosis, like when I looked at the studies, it did show the transcriptomic regulation of like different experimental conditions, but I think that they need to go in and look at aortic stenosis patients now to really find that, because the information known is that like the regulation of the protein functions in general, not like from the beginning, from the transcription step. That's it, yeah.
Yep, and that's a great analysis. And sometimes what happens is like at the transcriptional level, something can be regulated, but at the protein level, it's not, and vice versa. Like sometimes the proteins don't change and the transcripts change. So it's really important to look at both. Thank you so much.
Welcome.
All right. It was very stimulating. All three talks were very good. The presentations were fabulous. Actually, in fact, like I'm learning from them. I'm learning from them. So yeah, I'm looking forward for tomorrow's session.
Very nice.
8 weeks, if they have done this much of work, I can't imagine if they are gonna be like future scientists, they can push the boundaries. Yeah.
Thank you, Dr. Bano.
Yeah, thank you, Nutan, for this really amazing opportunity. So, I'm really happy that all of my mentees have done very well and put in the hard work. And it has been a great ride. And I would like to really thank Nutan for having this platform and bringing people together to, actually achieve something that's more than our individual efforts, right? So it's something that has come out as a result of Nutan's vision. So thank you very much, Nutan.
Could I add one more thing? I definitely want to thank you, Nutan, for this opportunity, because I always look forward. I'm a postdoc in a cardiovascular lab, and I always look forward to training undergrads, and we get a lot of high school students through the NSF, so National Science Foundation. And this year, because of the COVID-19 situation, I lost that opportunity, and I always look forward to it in summer. So this was perfect. So thank you so much. And all the students did amazing.